everyone, I am April Jane S. Bento from Bsed Sciences, and I am assigned to report to you the Trichinella Spiralis under the nematodes group. What is Trichinella spiralis? Trichinella is a nematode or cold roundworm parasite. It possesses the capability of infecting a wide range of mammals including pigs, horses, reptiles, and birds, but it causes disease only in humans. The life cycle of T. spiralis. Adult worms and encysted larvae develop within a single vertebrate host. An infected animal serves as a definitive host and potential intermediate host. A second host is required to perpetuate the life cycle of Trichinella. Domestic cycle most often involve pigs and anthropophilic rodents, but other domestic animals such as horses can be involved. In a sylvatic cycle, the range of infective animals is great, but animals most often associated as sources of human infection are bear, moose, and wild boar. The commonest Trichinella spiralis is found in a variety of omnivores and carnivores, including rats and pigs. Unusually, the vertebrate host acts as both a definitive and intermediate host. The adult worms live within the columnar epithelial cells of the small intestine walls. The larvae insist in striated muscle and the same mammal. Trichinella spiralis is the smallest known nematode parasite of humans. The males measure about 1.4 mm to 1.6 mm in length, and the females are 2.2 mm in length. The T. spiralis is a parasite of carnivores with a wide host range. Humans become infected when they eat undercooked meat, often pork or beer. Adult worms live in the intestine for up to 2 months, and the female produces larvae which migrate to skeletal muscle where they develop and encyst. Trichinella spiralis is a zoonotic infection acquired by ingestion of contaminated undercooked or raw or game meat. Infective cysts in striated muscle tissue are digested, releasing L1 larvae which mature into the adults in the small intestine. The adult forms release newborn larvae in a few weeks. As a result, it was discovered that Trichinella spiralis is in some facets more genetically similar to other multicellular organisms within the Animalia kingdom than it was to nematodes. Trichinella spiralis is the smallest nematode pathogenic to humans, is present on all continents of the world except Antarctica, and has been found in 55 different countries across the globe. This zoonotic parasite causes systematic trichinosis, a gastrointestinal disease spread by the consumption of raw meat, specifically pork. The earliest known case of trichinosis was identified from an Egyptian mummy from 1300 BCE, depending on the severity of the infection and location of the parasite. Victims may be asymptomatic or they may be experiencing diarrhea, pain, vomiting, swelling, fever, and rashes. Pathogenesis or the transmission of T. spiralis. Transmission of Trichinella spiralis only occurs through the consumption of animal meat infected with pathogenic cyst encasing Trichinella spiralis larvae. Natural hosts of the pathogen include rodents, bears, dogs, and even horses. Animals in close contact with rodents, such as domesticated hogs, are at the highest risk of contracting the infection. This is the pathogenesis or the transmission illustration. The larvae are ingested in raw or undercooked meats, and then the larvae are released from narcells and stomach. Larvae enter small intestine, which the adults mature and live in small intestine, and then the female sheds newborn larvae that enter lymph or blood. In its pathology is the heart failure and CNS damage. Newborn larvae are carried throughout bloodstream, and then the newborn larva enters skeletal muscle cell, and then the narcell larva complex form, which the larva matures in muscles, and then the last one is the normal or red and calcified narcell larvae complexes. As a result of persistent Nature of pathogen. Human contraction of trichinosis is generally attributed to consumption of raw or undercooked domesticated swine infected with Trichinella spiralis larvae. Human-to-human transmission is not possible unless ingestion of contaminated human meat occurs. The epidemiology of Trichinella spiralis infections is more common in Europe, Asia, and Southeast Asia. This disease is now endemic in Japan and China. Outbreaks are generally linked to cultural dietary preferences. In the late 1940s, the CDC reported 400 cases of trichinosis yearly based on statistics from 2010. The number of cases yearly has dropped to 20. This change can be attributed to increased awareness of the pathogen as well as strict regulations enacted about meat preparation as well the adoption of safer pig raising. The virulence factors. The virulent nature of T. spiralis can be attributed to the capsule that surrounds the larvae during transmissions as well as the high number of larvae female produces in the intestine. The capsule is believed to be an adapted advantage of the pathogenic worm, allowing survival in high temperatures up to 70 degrees Celsius. The capsule also protects the nematode from freezing temperatures for extended time periods as well. Clinical features. Severity and signs of symptoms vary on the number of larvae ingested by host. If the infection is minor based on the low amount of larvae ingested, Trichinella spiralis may not be able to colonize and the immune system can free the infection. If infectious dose is large enough within the first week following infection, gastrointestinal problems may arise including diarrhea, vomiting, cramps, or abdominal pain. Trichinella spiralis diagnosis. During first week, stool examination. Second week, peripheral blood sample of migrating larvae, and during third week, muscle biopsy and the muscle examined by compression, sectioning, and digestion. Trichinosis is endemic in human and pigs. Low source country and limited lab capacity. Most recommended method: slaughterhouse, trichinoscopy or digestion method, and the farm survey in pigs. This is the graph of reported cases of the Trichinella spiralis by CDC and their surveillance period of years. The black color shows the pork products, the gray colors represented the non-pork products, and the white color represented the unknown. How to treat the Trichinella spiralis? Moderate to severe infections required medication. Within the first week of infection, the main goal of medical intervention is to limit the spread of the larvae, which would lead to a systemic infection. Patients are generally administered an albendazole, mebendazole, or thiabendazole, an antiparasitic medication. Antiparasitic medication is the first line of treatment for trichinosis. Next is the pain relievers after the muscle invasion. And lastly, corticosteroids or cause allergic reactions when the parasite enters muscle tissue or when dead or dying larvae release chemicals in your muscle tissue. Lastly, how to prevent Trichinella spiralis. Due to meat being the main vector of transfer, meat preparation must be monitored and controlled. This would require careful preparation of meat as well as animal feed, which would include heating to high temperatures to ensure larvae death. Thank you for watching and listening to my presentation about Trichinella spiralis. I hope you learned something from my report. Thank you very much. Bye!